All right, so let's move on now to the second part of the talk, which is going to concern the discovery of the virus of KS itself. Where did the notion that KS might have an infectious precipitant come from? Interestingly, it didn't come from any microbiologist or virologist. It came rather from clinicians and epidemiologists who were studying the natural history of KS. And that natural history revolved around several striking facts. Of course, when the AIDS epidemic happened and KS went from being an extraordinarily rare tumor to a tumor that affected perhaps 20 to 25% of young gay men with AIDS, everyone initially presumed KS itself might be the cause of AIDS. But this idea soon collapsed. Of course, we know that KS occurred in Africa and the Mediterranean prior to the onset and spread of HIV through the human population, and that it could be shown that in classical KS there was no evidence of HIV in the lesion. Also, it was soon shown that in AIDS-related form there was no HIV DNA or RNA in the spindle cells themselves. Most KS biopsies didn't have any HIV-positive cells in them. So right away it became clear that although HIV was a very important cofactor that put people at risk for KS, it probably wasn't itself the cause, the proximate cause of the tumor. Another and particularly striking feature that supported that idea was that if you look among HIV-positive patients themselves, KS was not uniformly distributed among AIDS patients. So even among people who had contracted HIV and had gone all the way to a severe immunodeficiency, they were not equally at risk for developing this tumor. That KS was largely the province of gay men with AIDS, 20 to 25% of whom in the early days of the epidemic would develop KS. Whereas if you looked at hemophiliacs who had acquired HIV from blood transfusions, KS was relatively uncommon in them, 2%. KS was also relatively uncommon in women compared to men. Most women acquired HIV either through drug use, needle sharing, or heterosexual contact with HIV-positive individuals. And women had a very low incidence of KS, even when they went on to overt immunodeficiency. And most impressively, children who acquired HIV by vertical transmission from the infected mom to the newborn baby, by transplacental or perinatal spread of virus, almost never got KS, despite the fact that they had a very accelerated course and rapidly succumbed to the immunodeficiency. So these things all put the lie to the simple notion that HIV itself was the proximate cause of KS. And when epidemiologists looked at this data, one thing struck them, which is, of course, the striking prevalence of infection among gay men suggested that sexual transmission of some other agent in the context of an immunodeficiency might account for this result, particularly if the agent was more likely to be transmitted homosexually than by heterosexual transmission. So those facts swirled around the epidemiology community for a long time before they caught the attention of microbiologists, infectious disease specialists, and virologists. Most people weren't sure what to make of these facts, and because of the strong bias that many molecular biologists have, that information from epidemiology is simply correlative and not powerful in any direct way, many people in AIDS research discounted these results or simply ignored them. But two people who did not are shown here. These are my friends Pat Moore and Yuan Chang, who were then at Columbia and are now at the University of Pittsburgh. And they, as we did, took these results very seriously and began a search for viruses in KS. And now, of course, simple attempts to cultivate viruses from KS didn't work, and they'd been tried before. So their approach was to use a very advanced genomic method that had been recently described by Michael Wigler of Cold Spring Harbor, a method called representational difference analysis, or RDA. RDA is essentially a form of subtractive hybridization, a souped-up form of subtractive hybridization married to PCR amplification. And it can be used to explore two different populations of cells, the DNA of two different populations of cells, looking for segments of DNA that are present in one but not in the other. That's the fundamental notion behind subtractive hybridization. And Pat and Yuan did this on KS tumors and normal cells, normal skin or other tissues from unaffected patients. And after much to do, they discovered from this procedure, from a single KS tumor, 2 small segments of DNA of about 100 to 200 base pairs in length, which when sequenced, which were unique to KS, not present in the surrounding normal skin, and when sequenced, had distant evolutionary relationships to genomes of known human herpes viruses like EBV, Epstein-Barr virus. They were also weakly homologous to a simian virus called Herpesvirus Family. So these were not EBV genomes, but they were related to EBV genomes, and they correctly surmised that this must indicate the presence of a new herpes virus in these KS tumors. And they rapidly went on to show that these small sequences could be used as markers of KS, and they were found not only in the AIDS-related form of the disease from which they were originally isolated, but all forms, including the classical forms, endemic forms, whether KS was HIV associated or HIV unassociated from any part of the world, KS tumors always had these DNA sequences in them. And this result was rapidly affirmed by many other labs, including our own. So now it's clear that these sequences define the existence of a new virus, a new herpes virus, of a human herpes virus. Prior to this, there were 7 known herpes viruses. Now there are 8. The formal name of the KS virus is human herpes virus 8, but most of us use the more informative name KS-associated herpes virus because it indicates the major disease state with which the virus is associated. Now, when the KSHV genome was completely sequenced, as it was by 1996, it became clear where it stood phylogenetically. So all of the human herpes viruses cluster into 3 families: the alpha herpes viruses, the beta herpes viruses, and the gamma herpes viruses. And this segregation is now defined by DNA sequences, but initially these groups were defined biologically. Alpha herpes viruses are viruses that reside or establish a latent infection in neuronal cells. Gamma herpes viruses are viruses that establish latency in lymphoid cells. So that was a huge surprise that this virus that was derived from a predominantly endothelial neoplasm actually sorted with the lymphotropic herpes viruses. And for a while there was confusion about this. People wondered whether this was not the right virus, just some sort of passenger virus. But I'm going to show you very compelling evidence that it is linked to KS. But this lymphotropism set off a search that turned out to be productive. Once it became clear that the normal reservoir of KSHV is in fact the B cell, students of AIDS and especially AIDS-related lymphomas began to look for the presence of KSHV in lymphoproliferative disease. And Ethel Seserman and her colleagues and others rapidly established that there are 2 lymphoproliferative diseases that are linked to KS. One of them is a rare lymphoma called primary effusion lymphoma. So some of you may know that because of their immunodeficiency state, AIDS patients get many forms of cancer, KS being the most common. Second to that is lymphoma, predominantly B-cell lymphomas. Many of these B-cell lymphomas turn out to be EBV related, but one of them is KSHV related. It's a rare tumor called primary effusion lymphoma. Now, unlike KS, this is a classical lymphoma. This is a clonal B-cell tumor. It's an unusual B-cell tumor because the bone marrow and lymph nodes are generally normal, and the tumor arises in serosal spaces, spaces that are outside of solid organs. The pleural space outside of the lung, the pericardial space outside of the heart, the peritoneal space outside of the intestinal tract. These essentially form ascites tumors in these cases, and they occur in patients who are have very advanced AIDS. They carry a very poor prognosis. And these cells that are growing already in liquid culture in these spaces are adaptable readily into tissue culture and grow readily in culture, unlike KS cells. The other lesion that was found to be linked to KSHV is a disorder called multicentric Castleman's disease. The initial paper on this was written by a man named Soulier in France, who discovered that AIDS-related multicentric Castleman's disease is very tightly linked to KSHV, that these lesions have KSHV DNA in them. Multicentric Castleman's disease, or MCD, is an interesting disease that's right, again, at the cusp between the benign and the malignant. It is a polyclonal process in which B cells proliferate. Patients develop very high fevers, big spleens, lymph node enlargement. They have sky-high levels of IL-6 in the peripheral blood. So in those regards, they look like they have some sort of acute disease, but these patients have a proliferative syndrome and they require chemotherapy for its control. So again, a rare disease, but these two diseases are B-cell diseases linked to KSHV. I'm not going to say anything more about them today, but just to point out that they're further support for the idea that the principal reservoir of KSHV is in the B-cell, and there are pathologic consequences to the B-cell infection. Okay. An important aspect of this was cultured primary effusion lymphoma, or PEL cells, were central to advancing the virology of KSHV. With our colleagues Mike McGrath and Brian Herndier in San Francisco, who had isolated several PEL cell lines from patients at San Francisco General Hospital, we showed that these cell lines, which retained KSHV DNA, could be coaxed to produce the virus particles themselves. So the B-cell lymphoma lines are latently infected. They have cryptic infection with KSHV, but if treated with 4-ball esters or other chemicals, we showed that they could undergo lytic reactivation and produce infectious virus particles, electron micrographs of which are shown in this slide. And that provided a great source of early antigens and virus for future work. So finally, we had a system for cultivation in which virus particles could be collected and used to infect other cells. But also these PEL cells, because they were infected, could be used as a source of antigen to develop a serologic test. I want to tell you about the serologic test, but I wanted to emphasize why we were interested in making a serologic test for KSHV. Because remember, at the time, the question was KSHV really the agent predicted by the epidemiologists? Was it really causally linked to KS? We didn't want to spend a decade working on the molecular biology of a virus that was not the causative agent of KS. So we and others, and many others, including Pat Moore, Thomas Schultz, Chris Boshoff, many other groups, Denise Whitby, began to try to develop, based on PEL cells in culture, serologic tests. And the one that we developed is schematized here. It's an immunofluorescence test in which the antigen were latently infected primary effusion lymphoma cells. We incubate these with the patient's serum, wash, and then come in with a second antibody, a FITC conjugated or fluorescinated anti-human IgG. And this is what it looks like. The nuclei of the cells light up with an antigen that we called latency-associated nuclear antigen, or LANA. And it's a very crude test. Those of you who do immunofluorescence know that it's an insensitive method. The sensitivity of this method was about 75%, 65 to 75%, but it is highly specific. The pattern of staining, punctate nuclear staining, was highly specific. Okay. The test is laborious. It all has to be done manually, but it was our first entree into developing a test that would diagnose KSHV infection or exposure to KSHV in a human serum. Nowadays, this kind of testing has gotten much more advanced. It's typically done by enzyme immunoassay, often for LANA with one or more lytic antigens thrown into the mix, and the tests have improved to a sensitivity of 90% and a specificity of 97%, although there's still no commercially available test for routine diagnosis. But using our first-order home-brewed immunofluorescence test, Dean Kedis, an infectious disease fellow in my lab, studied over, in the aggregate, 5,000 patients from various groups. And his work and the work of others established a striking fact, which is that KSHV prevalence in the population strikingly mirrored KS risk. So if you looked at, as Dean did, at the blood donor population, this is a proxy for the whole population, only 2 or 3% were positive in this test. Similarly, low rates were found among HIV-positive hemophiliacs, and only slightly higher rates were shown in HIV-positive women, groups that we know were at very low risk for KS. But when he looked at HIV-positive gay men, seropositivity rates rose to anywhere from 30 to 60%, depending on the population and the test. And this is a group where we know roughly 20% of these folks had or would develop Kaposi sarcoma. So there was a striking segregation of KSHV infection or exposure with known risk for KS. And this was a very powerful fact that strongly advanced the argument that the genome discovered by Moore and Chang really was the agent that had been predicted by the epidemiologists. There's a lot more in the decade that has followed. Many groups now have examined populations all over the world, and what was true in the United States is true globally. Where KS is uncommon, the seroprevalence of KSHV is low. So in the US and Western Europe, it's somewhere between 2 and 7%. Whereas in the endemic zones of the Mediterranean basin, Sicily, southern Italy, Sicily, Sardinia, between 20 and 25% of the whole population is seropositive. And in Central and Southern Africa, where KS has long been known to be a highly prevalent tumor, the seroprevalence is between 50 and 70%. So again, not only in our domestic population, but globally, where KSHV infection is frequent, KS is common. Where infection is rare, KS is rare. The other thing about these low prevalence and high prevalence zones is they define two different epidemiologic patterns of KSHV spread. And the mechanisms here are still not fully understood. But in Western Europe and America, where seroprevalence is low, infection is principally derived by sexual transmission in adulthood, and especially among gay men. That something about homosexual sexual congress appears to be a particularly efficient way of spreading KSHV, and this is unusual in sexually transmitted diseases. Most of them have relatively equal rates of exchange among the different sexes. But in Western Europe and America, that doesn't seem to be the pattern. In the endemic zones, however, in Sicily and Sardinia, and especially in Africa, there is very efficient spread by some non-sexual route. We know this because many children, many prepubertal children in these cultures, seroconvert in the first decade or two of life before the onset of sexual activity. And then, of course, after the onset of sexual activity, rates of infection continue to rise. So there is sexual transmission in those societies, but there's also another form of transmission, which we believe is due to salivary exchange. There's lots of KSHV in saliva. Saliva is the principal body fluid that has virus in it, and it's thought that in those cultures, salivary exchange is an important route of transmission. Why these patterns are what they are, however, we really don't know. Why some regions display one pattern of spread and others another is something that remains to be understood. Okay. However, we did show with Jeff Martin, a very gifted epidemiologist here at UCSF, that among gay men, sexual transmission is very strikingly efficient and is strongly correlated with the number of sex partners. So here you can see the rate of antibody positivity rising as the number of sexual partners increase. And this was the first evidence for sexual transmission in the United States. All right, so let me sum up the epidemiologic argument that links KSHV infection to KS, because I think there's a nuance here that's very important to understand. Okay. So here are the points. First of all, KSHV DNA is present in all KS lesions, irrespective of whether they're HIV positive or HIV negative. Second, as I showed you, both locally and globally, KSHV seroprevalence tracks strikingly with KS risk. I haven't shown you this, but it's true that infection with KSHV precedes KS development and predicts an increased risk of KS. So in prospective studies that we did with Jeff Martin, we showed that if you were a young gay man living in San Francisco and you were dually positive for KSHV and HIV, and you received no effective treatment for either virus, over the next 10 years, 30 to 50% of those men developed Kaposis sarcoma. Finally, we note that KS is never observed in the absence of markers for KSHV. So if you do not have KSHV infection, you will not get KS. So those are the hard epidemiologic facts that link KSHV to the tumor. But it's important to realize that what they establish is that KSHV is necessary for the tumor. There's good reason to believe that KSHV infection is not sufficient for the tumor. I told you that somewhere between 3 and 7% of the US population is infected with KSHV. But these subjects in general have a very low risk. The actual magnitude of this risk hasn't been measured because KS is a relatively rare tumor outside of the AIDS population in the United States. But in Sicily and Sardinia, where island populations where there's very good epidemiology and where seroprevalence is 20 to 25%, there are pretty accurate numbers that allow us to estimate that if you're KSHV seropositive and don't have HIV, your risk of developing, only 1 in 3,000 to 1 in 10,000 of those people will develop KS. Okay. Therefore, it's very clear that in addition to KSHV, some other cofactor must be required. Now, we don't know what that cofactor is in the HIV negative form of the disease, but in the AIDS-related form, we know that that cofactor is HIV. Here's one illustration of that fact, and this is derived from epidemiologic studies of Jeff Martin back in the 90s. Before there was effective treatment for HIV, it was shown that your risk of developing KS prospectively was strongly linked to the viral load. So if you had greater than 1 million copies, 100,000 copies of HIV per mil of plasma, people rapidly and efficiently developed KS, whereas if you had an intermediate level, 10,000 to 100,000, your risk was slightly lower. And if you had less than 10,000 copies of HIV per mil of blood, spontaneously now without treatment, you had a lower still risk of KS. So that was one thing. There was a clear relationship, a dose relationship between the activity of the HIV infection and the risk for developing KS over 10 years. And a particularly striking feature has been what's happened to the epidemiology of KS since the introduction of highly active antiretroviral therapy, or HAART. Highly active therapy became widely available in the mid-90s, around 95, 96, and since then has been progressively adopted. And as HAART has become more prevalent among HIV patients in the United States, the incidence of new cases of Kaposis has plummeted, has decreased about 90%. So the data I'm showing on this slide are from Washington State, but the same thing has been seen all over the country and in fact everywhere where HAART has been widely adopted. So not only is there a correlation between spontaneous HIV replication and KS risk, but the interdiction of HIV replication has led to a striking reduction in KS risk. So all of this goes to say that KSHV is the essential driver of the process, but it requires an amplifier like HIV in order to produce clinical disease. All right. So how does KSHV infection predispose to KS risk? That's the question now. Nobody doubts that KSHV infection is the central element of pathogenesis here, but how does it predispose to KS? Of course, to a virologist, this question boils down to one thing. What viral genes are expressed in the tumor, and what do they do? And this is where, and we'll explore this question in the next part of the lecture.